All right, guys, so welcome to week 2 lecture of microbiology. I will focus on chapters 3 and 4. Chapter 3 has a primary focus of the microscope, so I will mention all different types of microscopy, how they work, how to properly and appropriately stain cells for visualization, and also a little bit about how we work with microbes in the laboratory environment. And then chapter 4 will cover basics of different types of cells, both prokaryotes and eukaryotes, and then we'll zoom in on the prokaryote. So for chapter 3, the microscope, things to remember here is that every microscope should have the ability to, number one, enlarge objects, and also the ability to show greater detail of that object. So every microscope has a specific magnification potential, and this is the way in which we visualize these forms of life on the single cell or the single organismal level, is by using a microscope to enlarge their appearance. So this would be magnification. Resolving power is referring to how much detail does the microscope actually offer. And as we're going to see, depending upon whether you're a light microscope or an electron microscopes provide not only greater magnification, but also a lot more detail. So again, magnification is just enlarging the size of an object. And magnification in most microscopes results from an interaction between visible light or electrons and the lens. All right, so I'm going to talk a little bit about the mechanics of how the scope works. Now, we worked with this microscope in lab if you're in the on-site course, but for the online guys, you have a different version of the scope. It's a monocular, so you don't have two ocular eyepieces, you only have one. But basic parts are the same. So you have the rotating or revolving nose piece with each objective lens. There should be 4, and the magnification potential is a little different on the monocular, but on the basic compound light microscope, it goes up to 1,000. So the highest magnification of this instrument is 1,000 times the actual size of that object. And usually at the higher magnification, you require immersion oil to make contact with the slide and the lens to provide for this greater resolution. So this would be the stage. These two pieces underneath here on the bottom is the iris diaphragm. So this is controlling the amount of light that is entering from the light source or the illuminator that is passing through the condenser, which is basically a series of lenses in here. And that will focus the light right onto the stage, onto the specimen. And then the light is passing through that iris diaphragm, through the condenser to the stage, and then through the objective lens. It then will pass through a prism in the compound light microscope and then to your eyes through the ocular. Now there's a couple series of refractions that occur here, and this is why the refracted light from the stage at this high objective, which would usually be on this scope 100x, the immersion oil would actually capture all of that light that's bouncing off of the stage. So because it's capturing all of that light, it provides for the highest magnification and highest resolution for this instrument. Now, like I said, you start off at the bottom of the instrument. This is where in a compound light microscope the source of the light is coming from. Again, it's going to pass through the condenser, hit the stage, through the objective lens, and then through the ocular lens to your eye. Now, one thing to mention here is that when you are looking through that ocular, whether you're using a monocular or one that has 2 eyepieces, that the real image is projected to the ocular, what you're looking through, where it is then magnified to form the virtual image. So you're actually looking at an inverted version of the real image or the real object. Okay. Now, when you're utilizing this microscope and all different types of light microscopy, please remember that the total magnification is equal to whatever objective lens you have set to times the ocular, which is always 10. Okay, so the ocular is always 10. If you're on 10x, then the total would be 100. So the total magnification of the final virtual image that you're seeing here is the product of the separate magnifying powers of two lenses, objective and ocular. Now, magnification is different than resolution. Magnification is just enlarging the object, enlarging the size of the object. Resolution is referring to the capacity to distinguish or separate two adjacent objects. So this is basically referring to the sharpness or the clarity of that image. And here's a decent example here to illustrate the principle of resolution, that the image on the right has a higher resolution because you can extract smaller bits of information from this image than the image on the left. So you can see, you can distinguish two adjacent objects that are closer together in this image on the right than the one on the left. And the actual resolution of the microscope that you're working with depends on the wavelength of light that is forming the image. Now, generally, with wavelength of light, the shorter wavelength is associated with an increase in the resolving power or a better resolution. Now, in order to quantify this resolution, the resolving power equals that wavelength, which I just said, shorter would give you a greater resolution. And this is usually in nanometers, over 2 times the numerical aperture. Now, typically the numerical aperture of the lens ranges anywhere from 0.1 to 1.25. And visible light is in the spectrum of 400 to 750. So usually for the oil immersion, taking into account this equation here, the resolution is around 0.2 microns or micrometers, and the magnification can range between 40 and 2000x. Now the basic compound light microscope goes up to around 1,000 times. And remember, like I said, at that immersion oil objective, which is 100 on the compound light microscope, I said that there's some light that is actually refracting off of the stage. The oil seems to do a good job of trapping those specific wavelengths of light. So because it's trapping more of the light is able to then travel up through that objective lens, through the ocular to your eye. So you're seeing more details about that specimen in the virtual image that is provided. Now, throughout the semester, whether you are in the online or the on-site version of the micro course, we will always be visualizing bacteria and any cell that is larger than that, but we do not have the magnification or the resolution potential with the compound light microscope to visualize something like a virus or a prion. So these are all examples of different bacteria, so Bacillus, Beryllium, Coccus here. And what we will be able to see is around 1 micron size will be resolvable with that particular microscope. Anything smaller than that we will not be able to see. So like I said, usually because of this unit of measurement for viruses and prions, you're approaching the nanometer unit of measurement. So 1 micron is workable when we have the compound light microscope. Now, there are all different types of light microscopy, so I just wanted to go over some basic characteristics of each type, and it's good for you guys just to be able to differentiate between the different methods. So bright field is the most commonly used method of microscopy, and the thing to remember about bright field is that you have bright light, it's a bright background, And this is really ideal for live and also preserved stained specimens. And again, anything that's larger than a bacterium. So what you're seeing here is actually a paramecium. It's a type of protozoan. But bright field allows us to visualize most microbes. Now, if you have a microbe that is translucent, transparent, has a very light color associated with it, and you want to look at that specimen unstained and not preserved, you may be able to do this using dark field, which actually involves a dark background rather than bright light in the background. So this will allow for a bright or a translucent specimen to be illuminated by this dark field. And this can be used for both live and unstained specimens. We also have phase contrast. So phase contrast is another type of light microscopy, and this is one that provides more resolution, a greater resolution or resolving power of the specimen that you're observing. And this is ideal for observing some of the intracellular structures within, this is again a paramecium, a eukaryotic cell. So some of the organelles you may be able to visualize using phase contrast. So we're still within the light microscopy realm here. We also have fluorescence. So this uses, rather than light in the visible spectra, this utilizes light in the ultraviolet spectra. And with this method, this has been useful in diagnosing infections. Typically, what we will do is we will use an antibody that is attached to a dye, so a fluorescent antibody attached to a dye that will emit visible light when it is bombarded with UV rays. So when it's bombarded by UV rays, if an antibody, for example, can bind to a unique antigen, so whatever the antigen is, if this is a particular strep bacteria that's expressing that antigen, the antibody will bind, and that dye, when you expose it to UV light, will fluoresce, and it will fluoresce the particular color that is associated with that dye, whether it's red, green. There are all different colors of dyes here, but this is used typically for diagnosing a lot of bacterial infections. And just remember here, it's in the UV spectrum. We also have scanning confocal microscopy. What I would remember about this is that much greater resolution than any of the others that I've mentioned thus far. This allows us to actually explore and visualize specimens on multiple depths or planes of the actual image. And this is possible through the use of a laser beam. So it's a laser beam of light that will scan the whole surface of the specimen and provide this greater resolution, shorter wavelength of light. And then we are moving on to electron microscopy, which has the greatest resolving power and the highest associated magnification potential. So unlike light microscopy, where we said it could go up to 2,000 times, you could magnify an image 2,000 times its actual size. Check this out, it goes up to potentially 1,000,000 times the actual size. So we can use electron microscopy to see viruses, which are much smaller than your average bacterium. And basically, it's a similar concept. It's just that rather than light, you're using a beam of electrons. And these electrons are made to travel in wave-like patterns when they're accelerated to high speeds. So remember I said that resolution correlated with a shorter wavelength of light. Now, these electron waves are 100,000 times shorter than the waves of visible light. So you have a shorter wavelength here with electrons, much higher resolving power. Now, as far as the mechanics of this, we said that for the compound light microscope, you have the light source, and then it's traveling through a series of lenses, right? So it passes through the condenser, the objective, and then through the ocular to your eye. Now, a little different here, the electron microscope, rather than a light source, you have an electron gun that will actually be aimed at and pass through, similar to light microscopy, a condenser lens, also through a series of objective lenses, an ocular, and then rather than your eye, onto a viewing screen. Okay, so similar mechanics here, but just remember, greater magnification, greater resolving power for electron microscopy. Now here's a couple particular pictures here illustrating the high resolving power. So I would remember there's two main types of electron microscopy. One is better, more ideal than the other. One provides 3D images of a specimen, whereas the other just shows you the image in one plane and indicates more dense areas of the specimen than others. So the darker areas here represent thicker, denser parts, and the lighter areas indicate more transparent, less dense parts. This is transmission electron microscopy. Now again, you can see some pretty small stuff. So you can look at, for example, a herpes virus or an Ebola virus, but with the transmission electron microscope, it is not in color and is not in 3D. This is different than the scanning electron, which provides these amazingly beautiful, detailed 3D images of a specimen. So scanning electron microscopy utilizes this, it's an electron mechanism here, but it will actually bombard the surface of the specimen after it has been metal coated. And so when the electrons are scanning back and forth over the specimen, it will produce this image, again, this virtual image of the actual sample that you're looking at. And this is color and 3D. So they're just fascinating. Some of these pictures are just awesome to look at. All right, so we have light microscopy and electron microscopy, two major methods that you should remember. Now, when you are actually ready to observe a specimen and look at it under the microscope, a lot of times you have to prepare it in a way where it is stained or preserved, and in this method you end up killing the cells, but for some of these cells you need to preserve them. For others, you can prepare what's called a wet mount or a hanging drop mount. This will allow you to look at live cells. Now, the advantage here is that if you're able to put together a wet mount, you could look at the motility. So this is really the only added advantage with a wet mount. You could see everything else on a fixed mount. You can see the size, you can look at the shape, the arrangement of the cells, and also an added advantage for a fixed mount if you're using a specific stain is that there are potentially differential or special stains that will show you specific structures that are unique to those cells. So I'm going to go over all of the different stains that exist. So there's two major groups of stains here. There's either a positive stain or a negative stain. And I would remember for a positive stain that these guys are basic, meaning that they have a positively charged chromophore attached to them. And the way that a positive stain works is that the whole surface of the microbe, because the whole surface of the microbe is negatively charged, it will be attracted to this positively charged dye. So because of that, the whole cell will turn a specific color. Okay, so if you're staining gram stain, for example, it's either going to come out pink or purple. You will actually see the outline of the cell pink or purple because you're using gram stain, the actual primary stain in that, as a cationic dye. This is on the contrary to an anionic dye, which is an acidic dye that will produce a negative staining result. Now, this is different than a positive stain because with a negative stain, you're staining the background. So if you're looking at a specimen using a bright field microscope, you're going to see, if you have a pink stain, that the background of the image is pink and the cells are actually remaining unstained. All right, I'm going to show you guys examples of this in a minute. Now, whether you are a positive stain or a negative stain, we also have different methods of staining depending upon what we are interested in observing. So if we just want to look at any cells that are in our hands that we're trying to prepare and look at under the scope, we just want to see what their shape is, what their size is, and how they're arranged. We could use what's called a simple stain. So this is just one dye. It's an easy protocol. So for the online crew, you guys are going to see that the simple stain will take the least amount of time. You use methylene blue. There's a couple washing cycles and you can look at it right away. If you're processing slides through a differential stain, this takes a little longer. There are more steps. But this provides you with specific information about what is present chemically in the cell wall of a particular type of bacterium. So Gram stain differentiates between gram-positive and gram-negative cell walls. Now, why do we care about gram staining results? It tells us what is actually in that cell wall. Is it a thick cell wall? Is it a thin cell wall? Do we have 2 cell membranes or 1 cell membrane? This is going to come up in a few slides, but these guys are really important. The gram stain, also acid fast. This is another differential stain. This can tell us whether we have acid fast positive or acid fast negative cells. And the thing to remember here is acid fast positive cells have what we call mycolic acid in their cell walls. Gram stainers do not. We also have endospore stain, which will specifically stain endospores, which are these dormant bacterial cell types. So we actually have uncovered endospores that are millions of years old. They are these dormant bacterial cells that can live on the verge of immortality, and you can actually take them out of the environment that you found them in and place them into an environment where nutrients are replenished and they will start to germinate and divide again. But a specific endospore is dormant at the time when it is formed. There are also structural stains, so these will reveal certain parts of the cell. So you can stain for a capsule, you can stain for a flagella. This is basically an extracellular exterior to the cell wall appendage that allows for movement. Or a capsule is basically an outer layer that provides protection, also is a virulence factor for attachment, and we'll talk about that in a few slides. So here's some examples. So you guys should for sure be able to identify, without having the labels up here, what these are. So just by looking at this, what I would remember is that all simple stains are blue. So methylene blue, that's what you're using to stain some of the cells throughout the semester. So it doesn't matter whether you have strep bacteria, whether you have mycobacterium in there, you have staph, they are all going to stain blue, okay, or E. coli. You'll see all blue cells. Now, if you wanted to differentiate between E. coli and staph bacteria, you could set up a gram stain. So a gram stain would stain E. coli cells pink and staph bacteria purple. Okay, so this is going to give you some information about the different types of bacteria that you have. And remember, I said acid fast is another one. And here's the spore stain. The endospores are actually in green. The cells are stained in pink. And then we have the negative stain. Remember, we said with a negative stain, you're going to stain the background and the actual specimen remains unstained. So this is actually staining for a capsule stain. So this is a negative stain. All right, so I now want to focus on the 6 I's of culturing microbes. So basically these 6 I's are referring to methods for how to study a microbe in the laboratory environment. Now, I would memorize all of these I's. So I'm going to talk about how to inoculate a microbe into a test tube or an agar plate or a culture to allow it to grow, and how then you would track growth over time. Also, if you had a mixed bag of microbes, how would you isolate those microbes? There's a process of incubation that would allow the microbes to grow. So this would be following inoculation. And then once they grow, how do we study them? So looking at the characteristics of their growth. What do they look like macroscopically? How are they growing on an agar plate? What do they look like microscopically? And then also information gathering and identification. Identification could be carried out via biochemical tests or a series of genetic tests. So there's all different steps involved in each one of these I's, and these are all the common I's that are used by every microbiologist when working in the lab. So these are kind of all of them in a little nice formulated chart here. I'm not going to read through every one of these, but I would spend a little bit of time on your own going over this chart and making sure that you understand what each one of these processes entail. So I'm going to give you guys a few characteristics of each one now. So isolation. This is a very important concept for you all to remember, is that when working with bacteria, and please remember, this is specific to bacteria when I say a colony. A colony is on an agar plate, which whether you're the on-site or online group, you will be working with agar plates and you will be looking at microbes growing on the surface of this agar. You will see a colony which is representative of, number one species, number 2, cells that have been derived from one single cell. So because of that, then there's a third characteristic here, number 3, they are all genotypically identical. So again, all of the cells in a bacterial colony are of the same species. They have all been derived from one single cell, and they are all genotypically identical. So if you have a mixed bag of cells, if you have a culture and you actually were to pour that culture or introduce a mixed bag of cells to the surface of an agar plate, you may just see a complete lawn of bacteria. So the whole cover of this agar plate, the whole surface of this agar plate, may just grow with a lawn of bacteria. Now, in order to actually observe different species, you want them to be isolated out into a colony. So how do you do that? So here we have a mixed bag of, it looks like there's 3 different species of bacteria. Now, if we look on this area of the plate, it looks like they're all mixed together, right? If you look down here, you can see that they've been separated out by bacterial colony. So this bright yellow bacterial colony is a different species than this cream-colored colony and this red-colored colony as well. But right here, if we were to actually pick up some bacteria from this area of the plate, we would pick up 3 different types. Okay, so how do you actually isolate and separate these guys out? Well, there's a few different methods, and the main one that I would like you guys to remember is the streak plate method. This is the quintessential technique for isolating bacterial colonies on an agar plate. So the way that this works is, let's say you have a sample of bacteria that has multiple species. You would introduce a little bit of that mixed culture onto one area of the plate. Okay, now I would suggest, even for the online group, when you guys are inoculating your plates, You can try to do this streak plate technique if you realize that you have an agar plate that ends up growing a ton of bacteria. You could take a sample from that plate and then place some on a new plate on one side of the plate, okay? So you have a little bit of bacteria here, but you don't have colonies yet. So what you're going to do is you're going to take a sterile stick or a sterile rod, and you're going to sweep and drag some of the bacteria in a few lines, okay, in a motion from this side of the plate to another area of the plate. Then you're going to take this stick and throw it out. Grab a new sterile stick, and you're going to drag from the edge of this bacteria, you're going to drag cells to another area of the plate. Then you're going to throw that sterile stick out and do it again. From this end, you're going to drag bacteria. Now, what the heck is the point of this? What we're doing is we're diluting the cells over time. So you start off here with maybe hundreds of thousands of cells. When you go through this zigzag motion, you are diluting to maybe 1,000. A third zigzag, maybe now you have 500. Hopefully, once you do 4 of these zigzag inoculations, you end up with one cell that has landed here, one cell that has landed here, and one cell that has landed here. That one cell divides to make 2. Those 2 divide to make 4. And you end up with, in the end, a colony that has thousands of cells from the same species, is genotypically identical, and has been derived from one cell. Now, you can do this either on a single agar plate using sterile sticks, or you could do this using the pour plate method, where basically, rather than diluting with a sterile stick, you're diluting with culture. So you could start off with a culture tube that has a saturated culture of bacteria. You could take 1 ml out of it, add it to a tube of sterile media, so you're diluting it 1 to 10, mix it up, take 1 ml, add it to other sterile media, so now you have a 1 to 100 dilution. And over time, if you plate this culture, you see that you have less and less bacteria and hopefully single colonies that show up. So the main take home here is that when you guys look at an agar plate, and for the online crew, when you guys set up these experiments, and then a day, 2 days, 3 days later, you go and you look at your plates and you see a colony, right? That means one single bacterial cell was introduced to that area of the plate by you, whatever you were swabbing it with, all right? Or also spread plate. You could do this with a sterile rod. They're calling it here a honey stick, but this is basically a glass rod that you place under a Bunsen burner. Usually you soak it in ethanol, isopropyl alcohol, and then flame it so it's sterile, and then spread the bacteria over the surface of the plate. So these are very important techniques in the lab. Once you actually have the colonies and you have the bacteria that has been grown, now we can inspect the species. We can look macroscopically on the surface of the agar plate. So what is actually growing? What is the pigment associated with these colonies? What is the shape of the colony? Does it have ridges? Is it concentric? Does it have a specific texture or pattern associated with it? There's also smells, different smells that are associated with different strains of bacteria. And whenever you're looking on your agar plate, you also want to look for contaminants. So there's a lot of times that if you're trying to work with pure cultures, again, if you just want one particular species of bacteria, you should only ever see one color of a colony, okay? Sometimes you can get different ones in here, meaning that you have more than one species, or you potentially have a contaminant. You have an unwanted microbe that's growing on that plate. This is probably going to happen a lot if you're not practicing sterile techniques. So the online guys, when you're setting up your agar plates at home, you need to make sure that you are keeping those plates closed, because there's stuff in the air that could fly in there and contaminate your experiment. Now, as far as identification, I would like you guys to remember the most accurate, definitive way to identify a microbe is usually by DNA sequence. This is how we profile and place microbes on the phylogenetic tree. So this is basically a tree that will reflect the evolutionary relationships or history of that particular organism. What is it most closely related to? So we always go to DNA sequence, and specifically it's actually the rDNA, the ribosomal DNA, or the particular genes that code for ribosomal RNAs and ribosomal proteins. We also look at the cells. So like I said, colony morphology. This would be macroscopically recording what is the pigment, what is the texture, right? What's the pattern of how it grows, the morphology, and also microscopic. So let's go back to this plate. Let's take one of these colonies off of this plate, introduce it to a slide, and process it through gram staining or simple staining or acid fast. And let's look at it under the microscope and see what is its shape, what is its cell wall characteristics. And also biochemically, this is another great way to figure out what microbe you have in your hands, because every microbe indeed has a unique metabolic profile, just like every organism that exists in nature. So we are all unique in that sense. And then also, immunologically, you can look at, this is basically just a fancy word for the antibody and antigen complex that you could test for. For example, if you are infected with a specific microbe that is displaying a streptococcal antigen, you could apply the streptococcal antibody to see if there's a positive reaction. So this would be an example of an immunological test. ELISA is an example of this. Even blood typing utilizes the concept of immunological reactions. Now, when you're working with microbes in the lab, how do we grow them? So you guys will be making agar plates and inoculating those plates with microbes, or we may be working with liquid culture. So the media that we're using to allow for these microbes to grow could be in liquid, also a semi-solid or solid on the agar. There are different types of chemical compositions of media. So there's synthetic versus complex. So synthetic has the advantage of it being chemically defined. So we know exactly what is in the mix, whereas complex has this different mix of extracts from various cell types, or meat from peptone, or mixes of different protein or amino acid recipes that we don't know exactly what is in there. And then every media that we work with in the lab has a functional type. So it can be used to grow almost anything, or we could select for a particular type of microbe, or we could use media to differentiate between different types. And there's all even more specifically for growth requirements like anaerobic oxygen requirements as well. So like I said, we can grow microbes in liquid culture, in a semi-solid slant, or on an agar plate, which would be in the solid form. And the benefit of agar is that it has this unique chemical property. So please remember this. This is super important about agar. Agar, when you heat it up, it turns to liquid. So you heat it up, it's liquid. You can add different chemicals, different antibiotics to it. You could even add the bacteria that you would like to work with to the agar when it's in the liquid form. And then you can pour it into a plate and wait for it to solidify at room temperature. So it will start to cool and solidify at 42 degrees Celsius. But once it completely reaches that room temperature is when it will turn to solid. And Another ideal characteristic of agar here is that it provides a framework to hold moisture for the microbes and also nutrients. And it is not digestible for most microbes. So this is the perfect ideal substance or compound to work with when growing microbes in the lab. Now, as far as media, nutrient broth and nutrient agar, basically the same thing, except one's liquid and one's solid. And these mixes or these recipes contain beef extract and peptone. The only difference is that you're adding agar. This allows it to turn to solid at room temperature. Now, remember I said there was a difference between complex and synthetic. So complex media contains at least one ingredient that is not chemically definable. So there's some stuff in there that we don't know exactly the chemical makeup of, whereas synthetic, we have an exact chemical formula. And generally what you guys will be working with is a lot of general purpose nutrient agar. We will be looking at and using some of the selective and differential media later in the semester, but I would say the first half of the semester, it's general purpose, and it is nutrient agar. Now, we have some media that contains complex organic substances, so we can add blood, hemoglobin, or special growth factors that are required by certain types of microbes. And I just want to say, since we're on this topic of growing microbes, there are a lot of microbes that, for example, from last week's lecture, I told you guys that there are billions of microbes that grow in the gut. A lot of those microbes we can't grow. We take them out of the gut and we try and inoculate them into all different types of enriched media or general purpose media, and they just don't grow. So for the ones that don't grow, trying to figure out what is it exactly that they need to grow can be a very tricky thing. So there are some that maybe even grow better in agar plates that have blood, for example. Now, in the case of an agar plate that has blood, this will actually allow you to identify if you have strep bacteria, because strep bacteria produce beta hemolysins, and beta hemolysins will lyse the red blood cells. So this actually can be used as a specific measure of identification or form of identification of this type of bacteria. Now, please remember there's a difference between selective and differential media. So if we want to select for one type of microbe, let's say you have a culture that has 20 microbes growing in it. It's a mixed culture, and you only want one of those to grow out of the mix. How do you do that? Well, you could use a selective media that contains one or more agents that will inhibit the growth of the other microbes and enhance the growth of the one that you would like to culture. So different types of this are mannitol salt agar, for example, different enteric agar, alcohol agar, MRS agar is another one for lactobacillus. This is different than differential. So for selective, you're selecting for one type of microbe, one species. For differential, this will allow multiple microbes to grow, but you can visibly see the differences among those microbes. So it's kind of similar to the differential stain, if you guys remember back to the gram and the acid fast, where if you have a mix of gram positive and gram negative, you could see the pink versus purple under the microscope. Similar here, you could see the different phenotypes on the surface of the agar plates, right? Purple versus red or white colonies. Whereas here, if it's selective, right, you're only seeing one. You're selecting for one type. And different types of differential here, MacConkey agar, eosin methylene blue is another one. But very important to remember the difference here. And then there's all types of enriched media or miscellaneous media. For example, fermentative tubes. that have media that contains sugar that can be fermented, and there's a little Durham tube measurement system in here that will tell you whether or not the sugar has been fermented and has produced a specific acid. Also, there's a pH indicator in here to show a positive reaction, and you can also see the gas bubble from carbon dioxide being produced. All right, so moving on to chapter 4. Chapter 4 focuses on characteristics of cells. So there are two basic cell types that exist on this planet that we know of: eukaryotes and prokaryotes. Eukaryotes being the larger, more complex cell type versus prokaryotes, which are smaller, they do not have organelles, and they encompass two whole domains of life forms. So both bacteria and archaic life forms are prokaryotes, whereas all other cells are eukaryotes. Now, both of these cell types share the common characteristics of a shape. They both have this internal content of cytoplasm surrounded by a cell membrane and also potentially a cell wall. They all must have DNA, so they have a DNA genome. In eukaryotes, there are linear chromosomes, and prokaryotes, there are circular chromosomes. They also must have these protein-making machines that we call ribosomes, and they must have their own metabolic capabilities as well. So remember, viruses we're talking about this semester are considered microbes, but they are neither eukaryotes nor prokaryotes. They are actually acellular. So they don't have these internal organelles like eukaryotes have. They do not carry out their own metabolism. Rather, they just have nucleic acid that is replicated to the future generation of that virus. And they also have a protein coat and some other characteristics that we will talk about. But two basic cell types, eukaryotes and prokaryotes. Now, both of these cells, not only do they have a metabolic potential and specific shape associated with them, along with this internal content, but they carry out all of the following processes. So they have the ability to pass on their genetic material from one generation to the next. And depending upon the cell type, it can be produced sexually or asexually. They also, all prokaryotes and eukaryotes, have the ability to absorb and utilize nutrients from the environment to grow and develop, to form a new cell over the course of cell division, or also to grow in size. Also metabolism. They have the ability to respond to outside or external stimuli, and also some may even have motility potential. They also have cell support, so along with the mentioned cell membrane, they may have rigid cell walls. These internal contents called vacuoles, granules, or inclusions. Not all organisms have these, but I will talk about those more specifically later on. And then they must have the ability to transport nutrients across cell membranes and also get rid of waste appropriately. Now, for eukaryotic cells, these encompass animals, including us, plants, fungi, and protists. So the fungi, algae, and protozoa, along with the helminths, those are all falling under this eukaryotic cell umbrella. And all of these contain membrane-bound organelles that are compartmentalized within the cytoplasm. They all carry out a specific function within that cell unit. And they also have a membrane that is associated with a nucleus, which houses the genetic material, so the chromosomes. Whereas for prokaryotes, all other cell types, those two domains, bacteria and archaea, and they're smaller and more simple, so they do not have all of these internal compartments. They do not have a nucleus. They do have ribosomes, and they do carry out their own metabolism, but the carriers for metabolism usually are found within the cell membrane rather than the mitochondria in eukaryotes. So for Chapter 4, for Week 2 lecture, we are going to zoom in on specifically prokaryotes, right? So bacteria and archaea are considered prokaryotic cells. So we're going to use a standard bacillus-shaped bacterium as the template for a prokaryote. So if this is a cartoon image of a prokaryote, this image is showing you the specific rod-shaped or bacillus-shaped structure. It has these outer layers, one of which is a cell membrane closest to the cytoplasm or the internal contents. And then as you traverse towards the exterior of the cell, you hit a cell wall, which all bacteria have a cell wall. This is critical and very important for you guys to remember. The only exception are mycoplasma, or some bacteria that will spontaneously lose a cell wall from exposure to antibiotics or other harsh conditions. But the only natural exception is mycoplasma. So cell membrane, cell wall, they also may have an additional external layer called a capsule or a glycocalyx, maybe even a slime layer. They also may have these external structures like fimbriae, which can be used for attachment, or a flagella, which will allow it to be motile. And if you look at the internal contents, basic structures that you should remember here is the nucleoid containing the chromosome, which is made up of the genetic material, so DNA. Also, these small, they're actually macromolecular structures, but these small little dots that you see here consist of ribosomes, which basically are the protein synthesizing machines within these cells. And then some other structures that you see here, like inclusion bodies or the filaments, the scaffolding within the cell. Also, a pilus, this can be used to attach to other cells, to swap DNA. So all of these structures we will talk about now in more detail. So external structures. Some prokaryotes may have flagella, which are protruding from the exterior of the cell, like you see here, a single flagella, or you could have multiple. You could have one polar flagella, you could have a flagellum on both ends, or you could have flagella all over the surface of the cell. But if it's not protruding from the external surface of the cell, then it may be internal. And if it's internal, these are referred to as axial filaments. You also may find fimbriae, which are these protein structures that are attached that you see here that typically function for attachment to surfaces or other cells within the environment, and also the pilus, pili is plural, and then that glycocalyx on the surface. Now, for the flagella allows the bacterium to rotate 360 degrees, and it will allow for motility of the cell through the environment. So, depending upon whether it's moving clockwise or counterclockwise will determine which way it moves. Now, also, depending upon how many flagella that you have, usually multiple flagella will move in the same direction to allow for that propulsion. Now, there are different ways in which the bacteria can actually move. So if it's counterclockwise, if the flagellum is moving in a counterclockwise manner, then that will result in a smooth linear direction, and we refer to this as a run. If those flagella are moving clockwise, then it tumbles, okay? So this is basically, the red is referring to a tumble, and the run is this smooth linear direction, which is when the flagellum are moving counterclockwise. Now, why would they be moving in their environment? In response to an external stimulus, either chemical, which we refer to as chemotactic movement, or a light stimulus, which would be phototactic. Now, those fimbriae that are all over potentially the surface of a prokaryote or a bacterial cell, these guys are very fine. They're made up of protein. So they're fine, proteinaceous, hair-like bristles emerging from the surface of the cell. And they can serve as what I'm going to call a virulence or a pathogenic factor. Now, a virulence factor allows the cells to cause disease, and virulence factors can increase the ability of a particular pathogen to cause disease. So in this case, E. coli, a pathogenic strain of E. coli that causes gastrointestinal symptoms or traveler's diarrhea, uses these fimbriae as a virulence factor to attach to the intestinal microvilli. So within the intestinal epithelial cells, it binds on here and causes the inflammatory response. It also may allow for attachment or adhesion to other cells similar to their own. Now also, some bacteria have what we call a pilus. I would like you guys to remember that this structure is actually only found in gram-negative cells. Now, the pilus is made up of these spiral-shaped structures referred to as pilin, and the pili functions to join bacterial cells for swapping of DNA information. So they can actually swap what we call plasmids, which are non-essential pieces of circular DNA. They can swap those pieces of DNA during this process called bacterial conjugation. So the pilus in gram-negative cells can contribute to the rise in antibiotic resistance because these cells can attach to one another and they can swap a plasmid potentially carrying an antibiotic resistance gene. So this increases the resistance levels and the virulence in gram-negative cells. Also, another virulence factor here noted is the glycocalyx. So this is a structure external to the cell wall, and it's made up of sugars and or proteins. So I'd like you guys to be familiar with the two types, either a slime layer or a capsule. Now basically they're very similar, but the main difference is that one is more highly organized than the other. So the capsule is the one that is tightly attached to the cell wall. So here we have a cartoon image of more loosely organized slime layer and more highly organized, tightly attached capsule. The glycocalyx, whether we have a slime layer or a capsule, has a function of, number one, protecting cells from dehydration or these drastic changes in osmotic pressure and also nutrient loss. And it also can function to inhibit the killing process by white blood cells. And this process is phagocytosis. So the glycocalyx can actually inhibit phagocytosis, which again can contribute to virulence or pathogenicity, the ability of the microbe to actually cause disease. Now, the glycocalyx also functions in the formation of these highly complex bacterial communities called biofilms, which we're just recently learning a lot more about how fascinating these structures are, because we think of bacteria as being single-celled organisms that exist solely on their own, so one single unit by itself. But if you actually study a particular community within a biofilm, you find that the bacteria actually on the chemical level are communicating with one another. So they're behaving more like a multicellular organism than a unicellular, which is super interesting. Now, this is a picture of the formation of one of those biofilms that I was just talking about on the surface of a catheter. So biofilms are a big deal, especially in the hospital environment. There are a lot of urinary tract infections that are caused from staph bacteria forming this tight network of staph bacterial populations on the surface of medical devices, medical implants, catheters, and also other inanimate objects or other surfaces, even the surface of your teeth. So if you slide your tongue over your teeth and it's slimy, that is a biofilm. So there's all different streptococcal bacteria that are living on the surface of your teeth, and they form this highly resistant layer. And if you don't remove them appropriately, they can lead to cavities or gingivitis. Now, another structure here, the cell envelope. So the cell envelope makes up both the cell wall and the cell membrane. Not all, remember, not all bacteria have both. There's one exception, and the exception is mycoplasma. So mycoplasma, we can cross off cell wall and just say mycoplasma has a cell membrane. All other bacteria have both. So these are the two layers that you would find covering the outside of the cytoplasm. And these layers maintain cell integrity. They regulate what is entering or exiting the cells. And we will talk a little bit more in detail about the cell membrane in particular. It really is a mosaic of all different types of proteins, channels, sugars that play a huge role in transport of molecules in and out of the cell. Now, for sure, with regards to cell wall, please have it ingrained in your brains to remember the details of the Gram stain. So Gram stain is something that you'll need to know for exams, for the final, for labs. Super important. Gram stain is a type of a differential stain. Remember, I just talked about this from chapter 3. And if you are a gram-positive bacterium, you will stain purple in the end. And what this means is that you have a thick layer of peptidoglycan. If you are a gram-negative bacterium, you will stain pink, and what this means is that you have a thin layer of peptidoglycan. Now, the other thing that's different here is that in gram-negative bacteria, there are 2 cell membranes, whereas in gram-positive, you only have 1. So hopefully this will sink in more when I show you guys an actual cartoon image of the gram-positive and gram-negative cell walls. Now, peptidoglycan is present in both gram-negative and gram-positive, just in different amounts. So this is actually the cartoon image of a chemical framework of peptidoglycan. And basically, peptidoglycan is made up of a repeating framework of long chain glycan chains here, made up of NAMs and NAG. So this stands for N-acetic muramic acid, or N-acetylmuramic acid, and N-acetylglucosamine. So you have these NAMs and NAGs, and these guys are linked by these interbridges. Basically, they consist of chains of amino acids. So you see glycine, alanine, glutamate, lysine, etc. Okay, so remember that. Peptidoglycan, it's a disaccharide. It's found in all bacteria, made up of repeating units of NAM, NAG, and also amino acids. So, who cares, right, whether you're gram-positive or gram-negative? Well, it's telling us a lot about the nature of that particular bacterial cell wall. So if you are a gram-positive bacterium, you have this very thick peptidoglycan layer, anywhere from 20 to 80 nanometers. So cartoon image here, the thick peptidoglycan is denoted by the brown layer, and the yellow is referring to the cell membrane. Now, gram-positive specifically will also have TA, or teichoic acid, and LTA, or lipoteichoic acid. Both of these will function in enlargement and also maintenance of the cell wall during cell division. They function to additionally move ions across the cell envelope, and also they will stimulate specific immune responses. So definitely another thing to remember here: teichoic acid, lipoteichoic acid, and gram-positive. Now, what's the difference between gram-positive, gram-negative? Gram-negative, remember, gram-negative has a much thinner layer of peptidoglycan. Gram-positive, thick, gram-negative, thin peptidoglycan. And additionally, we have two cell membranes. So we have a cell membrane surrounding the cytoplasm, so the internal contents, and an additional cell membrane on the outside of the cell. Now, unlike gram-positive that have the teichoic acids, gram-negative guys have LPS. They have lipopolysaccharides. And the key thing to remember here with the LPS is that the lipid portion actually can become toxic when it is released as an endotoxin during the infectious cycle. So gram-negative bacteria that are pathogenic may release this virulence factor, which here we're calling an endotoxin. So when you get sick with a bacterial infection, sometimes it's not just the presence of the bacterial cell itself, but it is the circulation or the presence of one of the components from that cell, like a toxin, an endotoxin in this case, or an exotoxin, which is a little different, but we'll talk about that later. Also, gram-negative cell walls may have porin proteins. So these function similar to ion channels that are regulating molecules entering and exiting cells. So great cartoon image for you guys here to pull up and study the gram-positive cartoon of a cell wall and the gram-negative. And you should be able to label and point out the different parts. So again, we're going to call this thick brown layer here peptidoglycan. We're going to call this a cell membrane, and we're going to say that teichoic acids are found in gram-positive cell walls. For gram-negative, you can tell right away because there's no thick layer here. It's thin, right? Thin peptidoglycan, two cell membranes, and don't forget the porin proteins and the LPS that could release that toxin. All right? So additionally, as far as the thickness of the peptidoglycan, I said that for gram-positive, it's 20 to 80 nanometers. It's a much smaller number here for gram-negative, 8 to 11. And the other thing I could point out here is that with regards to the periplasmic space, so this is basically the space in between the cell membrane and the cell wall. It's super narrow in a gram-positive. In a gram-negative, remember, you have two cell membranes. It's more extensive here. And usually these guys are less penetrable. So there are a lot of antibiotic-resistant strains that we're seeing are gram-negative. So especially for cell wall synthesis inhibitors, those drugs, those antibiotics don't seem to work as well. Now, as far as the steps of the gram stain, so remember, it's differential, right? So if you have a mix of staph bacteria that are gram-positive and E. coli that are gram-negative, you can process them through this staining protocol, look at them under the scope, and you'll see the gram-positive guys are going to be purple, the gram-negative will be pink. Now, this particular stain is very important. Classically, it's been used for bacterial classification and identification, and also diagnosing infectious disease and potentially guiding antibiotic drug treatment. So I would like all of you to remember the unique steps of this staining protocol. So remember, there's 4 unique steps. So again, if we have 2 types of cells, gram-positive and gram-negative, and we're going to process them exactly the same way, it's differential, meaning in the end, you'll be able to differentiate or distinguish between the pink gram-negative and the purple gram-positive. So what are the steps? Step 1, you're going to apply what we call the primary stain, which is crystal violet. What happens here is, due to the thick nature of the peptidoglycan found in the gram-positive cell wall, and also the peptidoglycan, the thin layer of peptidoglycan in the gram-negative, both cells will stain with the dye. So at this step, they are both purple. Now, second step, you're adding grams iodine, which is a mordant. A mordant basically creates a coordination complex between the dye and the cell. The dye will be trapped into the cell, and in this case you see here both still are purple. The third step of the protocol is to add a decolorizer. So this is where we start to see differences, and it has to do with the thickness of the peptidoglycan. So because it's so thick here, when you add alcohol, the primary stain does not leak out. The crystals will remain. But because you have a thinner layer, the outer wall is weakened, the cell loses the dye, and then following this fourth step where you're adding what's called a counter stain, in this case it's safranin, the gram-positive is not affected. The gram-negative will actually retain and take up that counter stain, and in the end will turn pink. So again, gram-negative pink, gram-positive purple. Remember that. Now also, note this exception. So I said all bacteria have peptidoglycan in their cell wall, okay? Except for Mycoplasma. Mycoplasma has sterols rather than peptidoglycan, and these sterols are actually found in the cell membrane. It can exist in many different shapes, so it's pleomorphic. There are also some bacteria that they have peptidoglycan, but they have this additional acid present, and that would be mycolic acid. So there are some cells that stain gram-positive that also will stain acid-fast. So if they are acid-fast, they will stain pink. Acid-fast positive cells stain pink. Acid-fast negative stain blue. And this is actually the basis for diagnosing, for example, Mycobacterium tuberculosis, which is the causative agent of TB or tuberculosis. Also, Nocardia has mycolic acid in the cell wall as well. These cause respiratory infections. Now, if we look at the cell membrane, I said earlier that the cell membrane is typically referred to as a mosaic, also fluid. So you can think of this layer constantly moving, or it's constantly fluid. It has all of these proteins, these ion channels, that are moving throughout the surface of this membrane. And it consists of a phospholipid bilayer made up of these amphipathic phospholipids. So they have these polar heads and non-polar fatty acid tails that are facing one another. And this cell membrane, which is found in both eukaryotes and prokaryotes, functions in providing the site for energy reactions. So unlike in eukaryotes where you have the ATP that is synthesized in mitochondria, ATP synthesis is a result of electron carrier function that's found in the cell membrane. Okay, so those cytochromes are found in the cell membrane rather than mitochondria in eukaryotes. Also, the cell membrane is important for nutrient processing, synthesis, transport, and the key is that it's selectively permeable, so it will only allow certain molecules to pass through or to leave that cell. Now, when you traverse through the cell wall, through the cell membrane, and you enter that cell, you will find the cytoplasm. So the cytoplasm is the gelatinous mixture that's made up of, majority of it is water. So water is the universal solvent. It serves as a solvent used in all cell functions. And it also contains sugars, amino acids, and salts as well. Now, as we're swimming around in the cytoplasm, we're going to bump into some prokaryotic cellular structures, such as the nucleoid. So the nucleoid is going to be the mass of genetic material, which is DNA. So DNA is deoxyribonucleic acid. It is a double helical structured molecule, and it's made up of these repeating trinucleotide repeats. So basically, DNA is made up of these 4 bases linked to a sugar and a phosphate, and those bases are A's, T's, C's, and G's. adenine, thymine, cytosine, and guanine. So you're going to find that sequence of A, T, C, and G within the chromosome, and that message is going to provide the directions to the cell to make all of the RNA and all of the protein that is required by that cell. So this DNA that you find in any cell, I don't care whether it's a prokaryote or it's a eukaryote, it is the software of life. In prokaryotes, it is going to be circular. Now, the one that is essential for the cell to maintain its life, its metabolism and reproduction is referred to as a chromosome. Bacteria are really good at swapping pieces of DNA that they don't need for survival. So these non-essential pieces of DNA that are also circular are referred to as plasmids. So it's an advantage, especially for molecular biologists, to exploit the use of these pieces of DNA for genetic engineering. So we can easily introduce a gene from a foreign species into a bacterial cell, and we can encourage that bacterial cell to make any gene product that we want as long as it's compatible. And there's a lot of really cool experiments going on with regards to genetic engineering in bacteria, and also yeast are easy to manipulate, and algae. Now, also ribosomes. So ribosomes are found in both prokaryotes and eukaryotes. In bacteria, they're a little bit smaller, but these guys are these structures that synthesize all of the proteins that a cell needs to carry out either structural functions or metabolic functions. So all enzymes are actually made up of protein. In the ribosome itself, again, the ribosome functions to synthesize a protein. It has two subunits. So it has a large subunit and a small subunit. And it has these little sites here, EP and A site, E for exit, P for peptidyltransferase, and A for aminoacyl. These are the sites where, if you guys have heard of tRNAs, the tRNAs will bind here and provide the code or the message for what protein, what amino acid should be synthesized and added to the linear polypeptide chain. But what I would really like you guys to remember is that they're smaller in bacteria or prokaryotes than eukaryotes. And each ribosome is made up of not only protein, but also RNA. So it's 60% ribosomal RNA and 40% protein. And they are found in all cells. Now, you also might find some additional internal structures like inclusions or granules. And the thing to remember is that these can serve as a mechanism for storage. And the bacteria can rely on the nutrients and the compounds in these storage bodies when they are depleted. And because of this, they vary in size, number, and also what contents are stored depending upon what species we are dealing with, what different type of bacteria we're talking about. Also, another very important internal structure which provides, you can think of this as a scaffolding, is the cytoskeleton. So this is an internal network, and it's made up of protein polymers. It's closely associated with the cell wall. So this kind of, again, this provides structure. You can think of it also as this little molecular highway that proteins can travel along to transport nutrients throughout the cell. And then you may also find this fascinating structure that we call an endospore. So an endospore is literally a dormant cell type. So it's not actively dividing, and an endospore may actually stick around for millions of years. So they have this long-term longevity cycle or survival associated with them. They're one of the hardiest forms of life. And they are usually formed, the endospores are typically formed in response to a harsh environment. So in an extreme of heat, drying, freezing, radiation, or chemicals where the cell would normally die, it will undergo sporulation, form an endospore, and protect its nucleic acid. And then when it's replenished, its environment is replenished, so you have the heat is taken away, you have restoration of water in the environment or less radiation and so on, you'll have the cell that will return to germination. So it will return to actively growing and dividing. Now, endospores are only formed in specific genre of bacteria. So you only find spores being produced from the Clostridium genus, the Bacillus genus, and Sporosarcina. Now, just for some application here or relation to disease, so Bacillus, if you guys have heard of anthrax or different forms of food poisoning, so Bacillus anthracis is the causative agent of anthrax. It can form spores. Same with Bacillus cereus. Clostridium perfringens, if you've heard of gas gangrene, is caused by that bug. Or C. diff, Clostridium difficile, which is antibiotic-induced colitis. This is another disease and another bacterium that can form these dormant cell types. And just remember, the endospore actually is characterized by two phases of its life cycle where it forms a spore and then it can start to grow again. So here's a picture of it where we're starting off with a vegetative cell. So let's say here's a happy Clostridial bacterium, and it's placed in this environment where there's very high levels of radiation. So the cell's about to die. So what it does is it will initiate this sporulation cycle before it dies. It will replicate its chromosome, it'll replicate its DNA. So this is actually the new spore that's being produced. This is not a form of reproduction. So we're not making a new cell. In the end, we end up with just one spore. We started off with one cell, we have one spore. We don't have two cells. Okay, so it's not a form of reproduction, but here we go. So we're starting the formation of the spore. It continues to expand and grow, and it will actively synthesize these hardy layers around the core of the nucleic acid. And the old DNA gets degraded and broken down, and it continues to form these outer coat layers throughout the course of development here that are very resistant to any extremes, again, in temperature or in the environment, one of the hardiest forms of life. And this thing can stay a spore for 10 million years if it wants, until it's placed in an appropriate environment where it can start to grow again. So they are dehydrated, they're metabolically inactive. We've found endospores that are up to 250 million, we've dated them back, 250 million years old. And they are highly resistant to ordinary cleaning methods and boiling, but we have found a way to destroy them. So especially for botulism, if you guys have heard of that, food poisoning, it's notorious in canned foods. We've found a way to process those canned foods in this autoclave cycle or using pressurized steam at a very high temperature to destroy the spores, and it seems to work very well. Now, a little bit about some morphological terminology here, or terms for shapes of bacteria. Please remember there's three basic shapes. We have a coccus, which is spherical, so it's round. We have a bacillus, which is basically, it's a rod, okay? Or we have a spiral, spirillum, which is helical, twisted rod. So some pictures here. So here we have bacillus, right? Here's the rod. Coccus, here's a spherical shape. Spirillum, here's the spiral. Spirochete or spirochete, these guys can move. Okay, so they have movement, they have motility, they have axial filaments. Also Vibrio, so if you've heard of cholera, Vibrio cholera, it's kind of shaped like a comma or a boomerang. And also some branching filamentous bacteria as well. So these are all shapes, but 3 basic shapes: coccus, bacillus, and spirillum. There are some, just to mention this concept, that there are some that can exist in multiple shapes. So you can see cells that exist in stacks of either bacillus or coccus, even though they're within the same species, or multiple arrangements as well. So maybe diplo or palisades, which I said was a stack of cells on top of one another. And some species are noted for their pleomorphism. So mycoplasma, for example, the one that I said does not have a cell wall, is a pleomorphic bacterium. So along with single cell shapes for bacteria, right? So I said coccus is one spherical shaped cell. We said bacillus, right, is the rod shaped. They also have these multicellular arrangements. So even though the single organism is one coccus itself, it may actually exist in nature in a chain of coccus. So if it's in a chain of coccus, this is what we call strep. If it's in a cluster of coccus, this is what we call staph. Or it could be in twos. We call those diplo. 4s, tetrad. Chains, again, would be strep. 8 would be sarcina. For bacilli, same thing. We could have 2 diplo. We could have chains, strep, or we could have palisades, which is a stack. Now, how do we classify all of the known prokaryotes? So we use the following information here. So we look at the microscopic morphology. We look at the macroscopic morphology, or the colony appearance. We also look at the bacterial physiology of the particular organism. And additionally, we look at serologically how the organism behaves, and sequence-wise, what is the sequence or the stretch of A, T, C, and G. Also, molecular analysis, particular gene expression profiles, for example. We look at the RNA, or we look at the proteins that are being expressed. Now, you can refer to this manual, Berge's manual. This has 5 volumes covering all known prokaryotes. And the definitive characteristic that is used for classification of bacteria is the genetic code. So we actually look at the ribosomal RNA sequence, and then we're able to place specific bacteria on this phylogenetic tree. And all prokaryotes fall into either the archaic domain or the bacterial domain. And there's 5 major subgroups within 25 different phyla. So as far as taxonomy, remember, this was from last week, right? The way that we actually name an organism is according to its genus and species. All genus species fall within a family. All families fall within a specific order. All orders fall within a specific class, and so on, phylum and kingdom. All right. So if we look at phyla for the domain Archaea, okay, you can see it's broken down by these 2 phyla, and then under this phylum you have the following classes, okay? And then under the class you would have specific orders, family, genus, species. All right, so the main thing just to know here and to remember is that the way that we classify prokaryotes is based on genetic information. It's based on the ribosomal RNA sequence. And prokaryotes are either in the domain Archaea, archaic, arcane for old ones, or bacteria, which are one of these 3 phyla: Proteobacteria, Firmicutes, or Actinobacteria. And these are the 3 domains that, well, 2 of which I just mentioned. So bacteria, Archaea, and then the third is Eukarya. So we're in here, And also the microbes that fall into this domain are the protozoa, the algae, the fungi, and the helminths. So those are all the ones that we will talk about this semester. Now, as far as diagnosing infectious disease, we can look at all of the characteristics that I mentioned before: biochemical, serological, taking a sample from a patient with a suspected infection and doing microscopic analysis, macroscopic analysis, and looking at the cell wall structure, the shape, how the cells are arranged, and physiological traits. Now, a few terms to cover here that are unique to bacteria, and this is with regards to differences in the species of bacteria, or different types of bacteria. A species is a collection of bacterial cells which share an overall similar pattern of traits, in contrast to other bacteria whose pattern differs significantly. So this is actually kind of arbitrary, right? So it's just saying that it shares an overall similar pattern. So usually, in order to be in the same species, you need to look very similar to all other bacteria in the same species as far as cell wall, membrane structures, internal contents, sequence, etc. Now, for a strain within a species, you may have a modified protein structure or a modified enzyme that participates in a particular metabolic process. This can also be referred to as a variety. Bacterial type can show differences in antigenic makeup. So serotypes, for example, or phage types with regards to the bacterial viruses that can infect those strains of bacteria. So the type, again, is referring to the antigen. And a good example of different serotypes would be in strep bacteria. There are multiple different streptococcal types based on the antigen that's expressed on the surface of the cell. Now, just to mention a few prokaryotes that have these unusual characteristics. So there are some free-living non-pathogenic bacteria, so some bacteria that do not cause disease. And also, surprisingly, we have bacteria that can carry out photosynthesis. So they can use carbon dioxide and light, specifically light, as a source of energy to produce molecular oxygen and glucose or sulfur. And these are actually the ones that produce molecular oxygen are referred to as blue-green algae, even though they're not algae, they're actually bacteria, but they are blue or green in color. And then also these guys, instead of producing oxygen, they produce sulfur, and then we also have the gliding fruiting bacteria. So again, cyanobacteria, blue-green algae, and these are gram-negative bacteria that can carry out photosynthesis. They produce molecular oxygen, or green and purple sulfur bacteria that produce sulfur. Or you have the gliding and fruiting bacteria that are gram-negative, and we won't actually work with any of these this semester, but these bacteria show this interesting growth phenotype where they will grow over moist surfaces, and they kind of grow into this fruiting bud at one end of the growth. And then lastly, these unusual forms of bacteria that were once classified as viruses. So we think of a bacterial cell as being able to exist on its own, and you could inoculate an agar plate or a culture tube with bacteria and watch it grow. You can't do that with a virus. So that's one of the characteristics of viruses, that they need host cells to thrive in. But these bacteria are exceptions. So rickettsias and chlamydias behave a lot like viruses. So rickettsial bacteria are actually obligate intracellular parasites. They're very small, they're gram-negative, and they cause diseases like Rocky Mountain spotted fever or typhus. Most of the species within this genus will cause infectious disease. Also, chlamydias additionally function as obligate intracellular parasites. And if you've heard of chlamydia, the STD, which is actually a very common STD, this is caused by Chlamydia trachomatis. It also can cause blindness or trachoma, and Chlamydia pneumoniae can lead to a respiratory infection as well. Now, lastly, just to mention a little bit about archaea, or the other prokaryotes. They constitute the third domain. And what's very interesting is, even though all of the cells within this domain are prokaryotes, they are more similar, they're more closely related sequence-wise, RNA sequence-wise, to eukarya than bacteria, which was a huge surprise when we actually were able to sequence that information. And they have this unique membrane lipid that's present. They do not have peptidoglycan. Okay, so different compounds that are found in the cell walls. What I would like you to remember is that all of these organisms live in extreme habitats. So you would find them living in salt lakes or hot springs or nuclear reactors. They're adapted to high heat, extremes of temperature, salt, acid, high pH or low pH, very high pressure, like barophiles 1 mile down in an ocean environment, and also extreme changes in atmosphere. And this includes methane producers, hyperthermophiles, halophiles, and also sulfur reducers. So that was it for chapter 4. So that concludes week 2 lecture. And next week we will transition into talking a little bit more about viruses. So I will see you guys next week.